Let me press this record button. All right, we're going to start up the spotlight session Monday, March 1st, 2021. Looks like we got a lecture exam, I think, this week, if I'm not mistaken. Hopefully we have some things that we want to talk about there. We're starting off in lab this week. We're starting off the axial skeleton. I just met tonight with my folks about basically the different structures of the skull and the features of the jawbone and the sternum and the spinal cord, lumbar portions. A lot of it's probably going to be just memorization at this point, really, when it comes to bones, at least pertaining to lab. So do we have any questions or do we have any topics that we want to talk about with lecture? I think one of our colleagues last night mentioned that we have the integumentary system tonight or this week. And what else did she say? I think the skin system, that's the epidermis, dermis, and hypodermis. Is there anything that we want to talk about tonight? Do we want to talk about metabolism? Do we want to talk about cell diffusion? I can revisit the action potential concept. I've been kind of going over that recently. In the past week or so. I'll be happy to hit on that again. I think students like that. So if we don't have any direct, okay, all right, cool. We got some stuff coming in. Study tips. All right, well, study tips. Study tips are what it really boils down to me is spending time with the material. Now that may be a cop-out answer, but what it is that when you spend time with the material, you listen to me, you listen to your lecture, you go over your notes, and you take we spend time with it, and you take continual passes at this material and learn it from different perspectives. Then eventually it's going to stick. Okay, so this is a class that you probably want to visit every day. You want to visit on it every day. If you visit the spotlight sessions and you come in and you have topics that you want to talk about, then you can obviously bring them out to me, and then I can try to explain it in probably a completely different manner than your lecture instructor. So that's kind of what these spotlight sessions are for. When I was in your all shoes, I wrote out all of my notes. I wrote them all out. And it takes a lot of time and a lot of effort, but I don't think sitting there and staring at your notes thinking you're going to memorize it's going to work. This is just too elevated material to just stare at it and think you're going to retain it, because your chances are you're not. So I recommend either recording your lectures or watching them again if you're online, and taking notes off those recordings, and then studying your notes and rewriting your notes. Really, and each time you rewrite it, you're taking different passes at the material, different attempts to learn it. That's what I kind of mean. And that's how I took my test grades from B's to C's and really into sustainable A's. So I think that's I think the key to success here. It's spending time with it every day. It's listening to your lectures, rewriting your notes in your own words, and then studying your own written notes or your study guide, so to speak. And all that really encompasses is spending time with the material. So I hope that helps. There is no quick fix. There is no cutting corners in classes like this, just because there's so much material and you have to keep up with it. And if you skip a day, if you want to take a few days off a week, that's fine, but you really want to revisit the material almost every other day or every day, just because it's a lot of stuff. So Brooklyn, I hope that helps you out with the study tips. Of course, let me know if you got any other questions. I'll be happy to answer them. All right, Jessica, interstitial growth versus oppositional growth. I don't know what that is, Jessica. What is interstitial growth versus oppositional growth? You may have to elaborate on that to help me out. I've never heard of that. Okay. Well, I mean, I may have to Google that and see what you mean by that. Oh, the bone. Okay. Okay. Like growth plate? I'm not sure if I can help you out with that. I may have to I may have to find the lecture material. Let me see if I can oh, wait. Huh. Okay. I've never heard of that before. That's why I'm literally looking at it right now as we speak. So it looks like, let me just read through it here. Interstitial growth produces longer bones as the cartilage lengthens and is replaced by bone tissue. All right. So I think, all right, when we grow, I think I know what we're getting at here. When we grow, babies, like for instance, like if babies are born very early, chances are their bones are not hardened very well, and it's more like a cartilage. Like sometimes you've seen little babies with helmets on because their skull is not fully developed. And what happens is that the cartilage will eventually grow, and then it layers out this kind of niche area of nutrients and kind of like a rich. Think of it as like a rich farmland or rich dirt, that helps foster good plants. It's the same thing here. It's like as we grow the cartilage out, it lays out this niche or this good nutrients of media or dirt, in this opinion, to help grow bones together. All right, that is interstitial growth. While oppositional growth occurs when new bone tissue is deposited on the surface of the bone, resulting in bone thickening. All right, so this is more like oppositional growth would be more like in the adult phase, okay? Now, this is probably pertaining to pathophysiology topics such as osteoporosis, all right? So that's obviously when bone gets degraded, right? It's the thickening or the density of the bone is lessened. That is what happens when osteoporosis happens. Sometimes that occurs when estrogen is postmenopausal women, when estrogen tends to decrease. Estrogen has a lot of protective measures when it comes to bone. And when estrogen drops off, parathyroid hormone can sometimes, and calcitonin hormone can get out of whack and start robbing calcium from the bones, and that's kind of where you get this lack of appositional growth. Like I'm talking when it goes away. Now, if we were to build it up, that would be a case where we're in the gym and we're lifting heavy weights. All right, this is something where this is over a longer period of time, where you have you're lifting more weights, you're putting more strain on your bones, and your body senses that. And then we have, if you remember, we have osteoclasts and osteoblasts, right? Osteoclasts break bone down. Class is like cutting, right? That will resorb a bone, as they call it. Resorb means take it away, versus osteoblasts, which is blast means build up or grow. So in this case, if we're in the gym and we're lifting heavy weights, our body will sense that. Our bones they'll sense the tension, and our osteoblasts will become turned on, and we will thicken our bone density and thickness by building on top of it, and that is called appositional growth. Yeah, Jordan, that's going to be the axial skeleton this week. So I will probably look over it tonight and send out an email. I hit the high points with my students tonight. We definitely looked at some stuff that I've seen in previous exams and quizzes. Now, if there are some real odd looking, real detailed points of the bone, like notches and fissures and sutures, then I will probably send out an email you check back in with me probably on Wednesday morning or something like that, and I can, or I think, yeah, Friday, it'll be done by Friday. But I think if you want to stop in on a spotlight session on Wednesday at 8 a.m., I could probably have a better, well, I'll tell you what, sit tight. Let me pull it up right here. I mean, why wait and do this? Let's take a look at the quiz. All right, so question 1 looks like we're going to be going over the sternum. It looks like questions 1, 2, and 3 are going to be coming from the sternum. Okay, so that's going to be including probably like the xiphoid process, the membranium, and then I think that's it. And probably the clavicular notch and the sternum and the jugular notch. All right, that's going to be the sternum. Number 4 is going to be a rib. It's going to be a question about identifying the rib, whether it's a true rib, floating rib, or false rib. Number 5 is going to be 5 and 6 are going to be coming from the cervical spinal cord, and it's going to be detailing you what just the shape of the cervical spinal cord. There's going to be a posterior view, and there's going to be like a side view of the cervical spinal cord, and it's going to want you to identify certain processes. I think, yeah, it's going to be either Atlas and Axis too. You'll see that in the lab. Okay. And then number 6 is going to ask you, I think it's going to ask you if that's a process or a cervical process or a tubercle or a dens. You'll see that in the lab. All right, 7, 8, and 9 are going to be more spinal cord questions. So it's going to be number 9 is going to be what is a foramen. Make sure you look up what a vertebral foramen are. So when you hear the word foramen, that really means like a cavity, right? Like a large hole. I think, Jessica, you were in there tonight, so you heard me hit on that pretty hard. That is the cavity, a large cavity. Now, it's not, like I said, if it was a cavity, they call it a cavity. This is like a large hole, like a gap. An instance for a foramen would be when you go over your lab, it is where the spinal cord meets where the spinal cord meets the skull, and there's a large hole in the bottom of the skull, and that's called the foramen magnum. That's not what's on the quiz tonight, or what I'm going over now, but it's an example of what a foramen is. And we see different foramen in the cervical spine area. So make sure you go over those too. So back on 7, 8, and 9, that is just more vertebra questions, excuse me. And I'm trying to think of anything else detailed I want to talk about. Now, one thing I want to tell you guys, when you look at the vertebra on, especially the thoracic and the lumbar, they're going to have animal features, as I call them. If you look at the side view of the vertebra, you're going to see that one of them, I think in this case, the cervical really doesn't look like an animal on the side. If you were to look at the thoracic and the lumbar, the thoracic looks like an elephant trunk. All right, the spinous process has like this long snout pointing down. That's called the spinous process. This is also seen in the quiz. And then the lumbar looks more like a moose, right? Instead of the spinous process pointing down, it's actually pointing out. Okay, it's like A moose spinous process or a lumbar spinous process is pointing outward versus a thoracic is pointing more like down. If y'all can see my picture here. So up, that's like the moose, I call it, and then down is the elephant trunk. Now you may be looking at me like I'm crazy, but you have to see it yourself. If you don't see it yourself, then you don't know what I'm talking about. I think some people, if you were in my class tonight, you probably are rolling your eyes because you've already heard this. So revisit that for other people. This is kind of what I teach my folks, how to remember or tell the difference between a thoracic elephant trunk and a lumbar moose snout. All right, that's just detailing the spinous process. And number 9 is some type of foramen. Basically, that's where the spinal cord comes through. And then number 10 is a picture of the sacral area of the spinal cord. Okay, so there's really no pictures of the skull whatsoever on this. So that's good because half the lab is over the skull, and it can kind of get hairy in the skull. So it looks like we spent most of our time on the cervical spine or the spinal cord bones and the sternum. All right. So how's that? Do you like that? That's pretty good stuff, right? Everybody should get a 10 out of 10. I pretty much just narrowed down your whole quiz into 3 different topics. I guess you guys are busy writing all this now. Yeah. I thought I'd get a thank you. Yeah, you're welcome, Jessica. I thought I would get a thank you for that. That's a huge gift right there. Yeah, anytime, Matthew. Loving it. All right. So, yes, rose belly. Yes, I think if I'm pronouncing your or Ross belly. I think if I'm pronouncing your name right. All right. So, is there any other questions that we want to go over tonight? We got a lecture exam coming up. If we want to go over some physiology, I can go over some physiology, some action potentials, and sodium and potassium, and depolarization. Yeah, anytime, Jordan. You can always ask questions. I'll be happy to go over some things. How are we on other topics? And Jessica, I hope I answered your question right on the interstitial growth and appositional growth of the bone. I've never heard those terms before. Okay, cool. I had to dust off the cobwebs on that one. So if I do want to go over the action potential real quick because it's a big deal and I do like going over that. I probably won't touch on the neuron anatomy. I think we kind of know what neuronal anatomy is with the shape of the cell body and the dendrites of the neuron. Let's go over the action potential, which is depolarization and repolarization. So when we have an action potential, we're sending out an electric signal, right? And this is how we measure membrane potential is we are placing an electrode inside the cell. So when we replace an electrode inside the cell, what happens is that we're measuring electrical conduction, right? We're measuring these cations and anions and these electrons, and if you get enough of them together, they create this kind of voltage, right? But you have to remember that we're measuring stuff in a cell, so we're really measuring at the mini voltage, the mini volts. We're measuring in mini volts is what I'm trying to say, not actual voltage, but mini volts. And when we have this, we have a resting membrane potential of probably minus negative 70 millivolts, right? Governed by potassium. So when a signal were to come in and activate voltage-gated sodium channels, as you recall, sodium moves from the outside to the inside. All right. So when we have that, we have sodium rushing in, which causes our membrane potential to skyrocket into the positive area. And that's called a depolarization of the action potential. And then at the top of the action potential, we eventually will open up what's called delayed rectifying potassium channels, where they're slow to react. And what that happens is when potassium opens up, or when these channels open up that are potassium channels, selective permeable to potassium channels, we have a concentration gradient that allows potassium to flow from inside the cell to outside the cell. Okay, and this is getting a large diffusion of positive cations from inside the cell moving outward, and we see a decrease in our membrane potential, also known as repolarization. Okay, so and then once we get at the bottom of the action potential, we see this like little dip, right? This little dip that kind of comes back and then stabilizes. That dip that kind of, when it goes a little bit farther negative and then comes back a little bit more positive, is called the hyperpolarization point of the action potential. Okay, and that is essentially what happens when we start to as you might recall, when we open up these channels, what really happens is that we're allowing things to go to equilibrium. And we don't really want that. We want to maintain concentration gradients. So when we maintain concentration gradients, we have to, well, when we get rid of the concentration gradients, because we're opening up all these channels and we're allowing things to equilibrate, we have to reestablish these concentration gradients. And how we do that is through the sodium potassium pump. That is the most important thing. That is if you recall, that's the active transport that helps reestablish these concentration gradients and reset for another active potential to come through. And so what happens is, if you remember, we pump three sodium ions for every 2 potassium ions. So 3 sodium ions get pumped out for every 2 potassiums get pumped in. And if you think about it, you may realize that, oh, well, we're pumping 3 out for every 2 in, and that gives us a net gradient, or a net difference of -1. And that's really why the action potential, resting membrane potential, the resting membrane potential is negative, because we have 3 sodiums going out for every 2 potassiums coming in. And that's why we have a negative resting membrane potential. So this is essentially what happens in the axon, right? And we have myelin that will help insulate the axon and help propagate these electrical signals down to our muscles or any other target tissue that helps move or carry out its job. Now, fast forwarding down to the neuromuscular junction is kind of where we have calcium that gets brought in, right? And so this is kind of what happens at where the neuron meets the muscle. And when the neuron meets the muscle, what really happens is that we have what's called the terminal bouton, right? And the terminal bouton is filled up with acetylcholine neurotransmitters and vesicles. And then we have a high concentration of calcium channels. And when the action potential arrives at the terminal bouton, this will cause voltage-gated calcium channels to open up, and then calcium will move in, causing the release of acetylcholine or neurotransmitters to release into the synapse or synaptic cleft. And that can carry all kinds of different hormones and neurons to fire, again, through acetylcholine, the neurotransmitter. Same thing as like serotonin and dopamine, but we'll save those for a different time. So I wanted to revisit that because that's kind of a pivotal concept that we got to understand when it comes to physiology. That is certainly my wheelhouse. That's what I like to discuss. Okay. Now, with that being said, do we have any other questions or concepts that we want to touch on? Do we feel okay on the lecture exam this week, I guess? if we don't have any questions, let me know, and so I don't have to sit here and wait. If you're still thinking, I can hang out. Okay. Matthew says he feels good. All right, that's cool. Does everybody else, are we good on the questions? Do we not have any more questions? Or are we still like scouring our notes? Okay. All right. So, okay. All right. Well, that's good. That's what I need to know. That's what I need to know. All right, good. Well, it looks like the majority of you guys appreciate that. It's better than you're staring at a screen. All right. Let me, if anything pops up, I will, let me go find the attendance form and you guys fill this out. And then after that, you're free to go. And good luck on the exam this week. And then if you have any questions, obviously please reach out. I expect 10 out of 10s on the lab exam this week. So good luck on that, but sit tight. So here is the attendance form. Feel free to fill that out and then let me know when you're done, and then I'll hang out and make sure everybody can get it figured out, and then I'll turn you guys loose. All right, sounds good. Thanks, guys. Good luck this week. Remember what I told you on the study tips. I think that really helps. If you're not happy with the scores you're getting, I know you can probably do better. You just got to tweak some things out. And so please remember my study tips. All right, you guys are free to go. Thank you. Hey, you're welcome, Haley. Yeah, anytime, Jacoby. You're welcome.